Last lesson, we looked at whether photosynthesis was happening or not, and we learnt the test for photosynthesis, how to test a leaf for starch to see if it had been photosynthesizing. Today, we move on to looking at how fast photosynthesis is occurring. This is known as the rate of photosynthesis. So how could we measure how fast a plant is photosynthesizing? Well, we'd have to look at what the plant is producing. Do you remember the two products of photosynthesis? Did you say glucose and oxygen? So just thinking about that, could you measure the amount of glucose made, how fast it makes it? That would be so difficult to measure how fast molecules of glucose are made. But it is possible to measure how quickly oxygen is made using plants that live underwater. The oxygen they make forms bubbles and they rise up. And so you can count the number of bubbles coming off in a particular period of time. We use pondweed. It can be something called elodea or another pondweed called kabomba. So here's a little film clip to revise what we know so far and also to look at how to measure the rate of photosynthesis.
What does this and this have in common with this? They're all feeding. Plants feed using a process called photosynthesis. Photosynthesis is the chemical reaction through which plants make glucose. It also produces oxygen. It is crucial to all life on Earth because it is the source of nearly all the food and oxygen we need to stay alive. But what do plants need to photosynthesize? First, they need water. Water is as essential to plant life as it is to us. It is one of the reactants in the photosynthetic reaction. Plants which have little access to water go to great lengths to conserve the water they collect. These desert plants survive by maximizing the amount of water they get from their harsh conditions. The other reactant needed for photosynthesis is carbon dioxide. Most plants collect this from the air. To do this, they have tiny holes in their leaves called stomata. As well as water and carbon dioxide, plants need one other thing for photosynthesis: light. This provides the energy for the chemical reaction to take place. Photosynthesis takes place in the chloroplasts in plant cells. Chloroplasts contain a green substance called chlorophyll, which absorbs the light energy needed to make photosynthesis happen. To summarize, photosynthesis takes the raw ingredients of water and carbon dioxide and uses light energy to make glucose and oxygen. This pondweed is called kabomba. Because it lives in water, we can see it releasing bubbles of oxygen as it photosynthesizes. When we vary the amount of light the plant receives, we can see that it affects the rate of photosynthesis. This one has full light, and we can see it bubbling vigorously. This one has been shielded from the light, and the photosynthesis has stopped.
So in the lab, if we wanted to measure the rate of photosynthesis, we could use a really simple setup like the one in the picture here. We have a lamp here, and it's producing light here, shining onto a piece of pondweed here. We actually have used Elodea and we've used Kubomba in the past. And the pondweed is sitting in water with a freshly cut end here, so that you can count the number of bubbles being given off in a certain period of time, like, for example, 1 minute. Now you'll notice the lamp is at a specific distance away from the pondweed. Here it's 50 centimetres. So you would do it with that first, with the 50 centimetres, and then you would move it closer to, say, for example, 40 centimetres away, then 30, then 20, then 10, and then right up to 0 centimetres away. And that would allow you to find out what happens if you increase the light intensity, the amount of light that the pondweed is getting. What happens to the amount of oxygen that is produced? You can have a think for yourself now. What do you think will happen? Now, a slightly more complex set of apparatus here, where you have a funnel over the top of the pondweed, and you have this inverted test tube. That's a test tube upside down, and it's full of the pond water. And as the pondweed bubbles in the presence of the torchlight here, as it bubbles, it produces the oxygen, and the oxygen can actually be collected in the test tube here, because as the oxygen rises up, it will push the pond water down and out of the test tube down here and into the bottom. So you end up with pond water collected here. This process of collecting gases is called downwards displacement of water. This is just another step in more complex, in that this time you are doing exactly the same thing. They haven't shown the light here, but you are measuring the rate of photosynthesis. This time you are actually measuring the volume of oxygen produced per, say, 5 minutes. And so you can actually get a measurement here, making it even more accurate. And this one here is probably the most accurate way of doing it. You can see here the light is again at a specific distance. This one looks like it's at, difficult to tell there, but about 40 centimetres away from the pondweed. And it will produce the oxygen here, which will travel up through this delivery tube here and be collected in a gas syringe. So as more gas is collected, this plunger part here will move backwards, and you'll be able to measure the volume of gas collected in a certain period of time, depending on how close you've got the light. But what we do in the lab is we use the most simple version here. So if you were in the lab, you would now be collecting these pieces of apparatus. You'd collect a lamp, you would collect a meter ruler here, which sits along here. You would collect a large boiling tube here and a piece of pondweed that you would cut a fresh end to, and then you would cover all of the pondweed with pond water. So you would turn the light on with the lamp at 50 centimetres, and you would measure how many bubbles. You would literally count the number of bubbles that come off the pondweed within a certain period of time. We usually allow 2 minutes. So the room would be completely dark, and when you were ready, you would start the timer and you would turn your lamp on, and you would count how many bubbles came off the pondweed within 2 minutes. You would then repeat this whole process, but moving the lamp 10 centimetres closer. So then it would come to 40 centimetres away. Once again, you time for 2 minutes, count the number of bubbles produced, and then you would repeat again at 30 centimetres, then at 20 centimetres, then at 10, and finally at 0 centimetres. You can see down the bottom here the results that we have obtained in the past, where the distance from the beaker is written here, and the number of bubbles collected in 2 minutes is written here. So I've copied in here some results for you as if you had done the experiment. And the next slide here is an evaluation of the experiment. Once again, I've copied in for you the graph here because you wouldn't be able to draw that online. But what you will be able to do is answer these evaluation questions here. So that's going to be your job in a minute. And I've attached both these slides, the one before here and this one, into a Google Slides document, and it's called Experiment to Measure the Rate of Photosynthesis. So in a moment, not quite yet, you're going to open that up and you're going to fill that Google Slides document in using text boxes. Just having a look here, one of the questions that it will ask you is, what is the problem with this experiment? I want you to think about what the lamp here also gives out as well as light. This is a problem. If two factors are changing, then we don't necessarily know which factor has caused the increase or decrease in photosynthesis. So have a little think. What else do lamps give out that you have to be a bit careful about with touching them that might cause the pondweed to photosynthesize faster? Thinking about this, and how could you prevent it? What could you potentially put in the way here that would allow the light to pass through, but not this other factor that would cause a problem? So that should help you with your next slide here in the evaluation, which you're going to do in a moment. But for now, before you do that, I've got a film clip here for you to watch and a little task for you to do.
You'll need a dark room with a desk lamp, some pondweed, a glass of water, a ruler, and paper and pen. To start, put some pondweed, which can be bought from any pet shop, into a glass full of water. Then, set up a lamp 10 centimetres away from where the pondweed floats. Give the pondweed 5 minutes to adjust to the lamplight. Start to count the oxygen bubbles coming from the pondweed for 1 minute. Water actually contains small amounts of carbon dioxide gas, which is dissolved in it. The plant uses the energy from the lamplight to turn the carbon dioxide and the water into glucose, which it needs to live. The byproduct of this reaction is bubbles of oxygen. After a minute, move the lamp back another 10 centimetres and allow the pondweed to get used to the more distant light. Then get counting the oxygen bubbles again. Do this again from 30 centimetres away. What you should notice is that as you move the lamp further away, the number of bubbles decreases. The closer the light, the more energy is going into the plant, which speeds up the reaction, producing more oxygen bubbles.